So I think we'll get going, everyone. It's a pleasure today that we're going to be welcoming Dr. Pieter Cullis to the University of Manitoba as our Gairdner lecturer. Before I start, I just want to acknowledge that the University of Manitoba campuses are located on the original lands of the Anishinaabe, Cree, Ojibwe, Cree, Dakota, and Dene peoples, and on the homelands of the Métis Nation. We respect the treaties that were made on these territories, we acknowledge the harms and mistakes of the past, and we dedicate ourselves to move forward in partnership with Indigenous communities in a spirit of reconciliation and collaboration. The Gairdner Awards Lecture celebrates the world's best biomedical and global health researchers. The Gairdner Foundation was established in 1957 with the main goal of recognizing and rewarding international excellence in fundamental research that impacts human health. To date, there's been 402 awards that have been bestowed on laureates from over 40 countries. And of those awardees, 96 have gone on to receive Nobel Prizes. In fact, many of the Gairdner lecturers, it's been seen now as almost like a pre-Nobel award. Today, our speaker is the recipient of a Canadian Gairdner International Award. There are 5 awarded annually to outstanding biomedical scientists who have made original contributions to medicine, resulting in an increased understanding of human biology and disease. Dr. Pieter Cullis is director of the Nanomedicine Research Group and professor of biochemistry and molecular biology at the University of British Columbia. He and his co-workers have been responsible for fundamental advances in the development of nanomedicines employing lipid nanoparticles technology, and he'll be talking about that today. He's co-founded 11 biotechnology companies, published more than 350 scientific articles, and is an inventor of over 60 patents. He has also co-founded 3 not-for-profit enterprises, including the Center for Drug Research and Development and the Nanomedicines Innovation Network. The 2 drugs enabled by lipid nanopore technology, delivery systems devised by Dr. Cullis and members of his UBC lab and colleagues in the companies he has co-founded have recently been approved. So with this, I want to also just highlight Dr. Cullis has had numerous awards, including the Order of Canada in 2021, and the Vin Future Prize and Prince Mojito Award in 2022. Peter, please give him a warm welcome.
Okay, thanks for that introduction, Peter. Okay, what I'm going to talk to you about today is really how accidental, in a way, science actually is, as opposed to the rather linear way that we often see it being portrayed. And so that's why I entitled it Science and Serendipity. There's a lot of good luck that's part of this, and that it relates to the lipid nanoparticles that enable the COVID-19 vaccines. I could also entitle it 50 Years of Lipids, because that's pretty much what it is. So I've really been involved in this field for rather an embarrassingly long time. Now, how do I get these to advance? That's the next thing. I'm just going to press a space bar here, and maybe that'll work. Okay, there we go. You've seen various public announcements about whatever analyses of the vaccine, the mRNA vaccines, and in some cases you see it's coming inside what people term as being tiny bubbles of fat. So this is a headline from Bloomberg, and this is where the mRNA is coated in lipids. And so what I'm going to talk about is really these tiny bubbles of fat. First of all, I'm going to say that they're not really tiny bubbles of fat, but anyway. So what are they? They're already tiny. We're talking about systems that are 100 nanometers or less in diameter, so 100 times smaller than a cell. But they're not really bubbles of fat. They're nanoparticles made of membrane lipids that have really taken my whole career, 50 years, to develop. So it goes way back to 1972 when I was involved in for 30 years I've been doing basic studies on membrane lipids. Starting about 1985, we started to apply some of that knowledge to development of lipid-based systems. We should have both of these things on, shouldn't we? Anyway, to development of systems to deliver cancer drugs, which is, it was what we started to do. We started off trying to cure cancer. Didn't quite manage that, but we've done a few other things. And then starting in 95, applying the knowledge, our basic knowledge, to deliver nucleic acid-based drugs. Now, just going back to the beginning, I got my PhD in solid-state physics in using magnetic resonance in 1972, and I was looking at semiconductors at 4 degrees Kelvin. This is like transistors on the moon. Anyway, I decided after doing this that I really had to do something different. Most of the interesting problems that I could see were well outside the field of physics, particularly in the life sciences. And so I got interested in applying NMR in the life sciences, and I got a fellowship to go to the biochemistry department in Oxford in '73, and I knew absolutely nothing about biochemistry. So this was quite a what's the word? Inundation by fire. Now, I knew a bit about NMR. I published my last physics paper in 1976. It consisted of about 96 equations. But anyway, that's pointing out that when you learn something as a physicist, you learn it in some depth. There's other things that you learn as a physicist. Basically, when I was an experimentalist, when you need an instrument that isn't available, you build it. And of course, going after basic problems is another aspect. Well, when I got to Oxford in 1973, they had big magnets that was one of the more powerful ones in the world at the time, actually. They had no electronics to go with it. This is me in Oxford. We had to build, me and a graduate student, we built all the electronics to go with this, to make this NMR machine actually work. So that's how I spent my first year there. Good job I had a physics background, otherwise it would have been a bit tricky. Well, it was a bit tricky as it was. And then I used the NMR machine to study lipids in biological membranes, so really basic stuff. Just going after basic problems. And so this is a model of the biological membrane, which was the Singer-Nicholson model, which is still quite relevant today. Showing the various components, proteins, and lipids that we're all very familiar with. Now, all biological membranes rely on a lipid bilayer as a structural element. So this is taking out the proteins. This is what you're going to see. And the membrane lipid, of course, are amphipathic. I mean, they have a polar head. They're self-assembling, which is a wonderful quality. They have one end that likes to be near water, and the other end that likes to be sequestered away from water. So the basic questions I got interested in were twofold. I mean, one of them was, well, biological membranes contain a lot of lipids that if you pull them out and then put them in water, they don't adopt bilayers. So what are they doing there? And what's their roles? So why are these non-bilayer lipids there? So it's called lipid polymorphism. A long way from vaccines, but we actually used the knowledge that we gained studying lipid polymorphism to develop more effective delivery systems for mRNA. And the other question that we looked at was lipid asymmetry. So the lipid composition on one side of a biological membrane is different than the lipid composition on the other side. And so we asked the question, well, can this be related to ion gradients, that are present across membranes, whether they're sodium, potassium, or proton or pH gradients? And so this is, I spent a lot of time and still do spend time actually on investigating those properties. And what we discovered was that we could use NMR, phosphorus NMR. I mean, phospholipids have a phosphorus in them. So we could use a phosphorus NMR to study these different structures that lipids adopt. Just add water, and these will form bilayers or these other structures. If they form a bilayer, then their main motional characteristic is rotation around the long axis. And so so this gives rise, due to the phosphorus and the phosphate, to this kind of characteristic line shape with a low field shoulder and a high field peak. So it's quite diagnostic of a lipid bilayer. On the other hand, if they adopt these strange structures such as the hexagonal H2 phase, which consists of long tubes of water surrounded by lipids on the outside, these are about, say, 10 nanometers or so or less in diameter, then they can, additional motional averaging, they can rotate, they can go around these cylinders very rapidly. This ends up giving you a different line shape with a low field peak and a high field shoulder. So really quite, and then if they had total motional averaging, then we'd see a very narrow line shape. And so this allowed us to study the properties of many lipids in terms of their phase preferences and how we could shift between one or the other. This is Ben de Kruif. I worked on a lot of this with him in the late 70s, early 80s, and just pointing out that we could rationalize this by, and this will come back in when we talk about the vaccines. The lipids in a bilayer structure having more of a cylindrical shape, the area in the head group region being more or less the same as in the acyl chain region, whereas lipids like this structure, such as these hexagonal phases, have a cone structure where the area per molecule in the head group is somewhat less than the areas attended by the acyl chains. So very simplistic thinking, but we then went on saying, okay, well, what are the functional roles of these lipids? Now, in order to investigate these roles, it's very difficult to do anything in the intact membrane because there's literally hundreds of different lipid species there. And trying to pick out the properties of one lipid versus others is pretty much impossible. And so you have to use very simple model membrane, or what's termed liposomal systems. Now, at the time, there was not really a good way of making these systems. We wanted to make these systems with one bilayer, then inside and outside, et cetera. So we devised a high-pressure extrusion technique where we basically took larger lipid bilayer systems and rammed them through a polycarbonate filter with a 100 nanometer pore size. And so out the other end, you get these unilamellar systems, just like this. So this is where building an apparatus, actually we formed a company on this that served us very well. But anyway, we could use these liposomes to show that membrane fusion depends on, if we have non-bilayer lipids and we add them, we put a small proportion in with these bilayer forming vesicles, then we can engender fusion between them, and we'll see. So they've definitely, and this and much other evidence indicates that membrane fusion is really dependent on these lipids. And of course, membrane fusion is vital to life in many ways. Now, we also use these liposomes to demonstrate the consequences of lipid asymmetry. And I'm going into this a bit because it points out the importance of doing quite basic research and how you can use the insight you gain from basic research to inform and it gives you tools that you can use for more therapeutic applications. And so we synthesized this ionized cationic lipid called DODAP. And the thinking here was that this lipid has a property, it's a tertiary amine, it can exist in a protonated state, which is positively charged, and that's at low pH, whereas at, say, neutral pH, it becomes reprotonated, and you have a net neutral molecule. And the net neutral molecule can diffuse across the membrane really quickly, but if it comes to the inside and gets protonated, it becomes positively charged, well, it can't get back out again. Because charged molecules don't get through lipid bilayers very easily. And so we could drive lipid asymmetry in response to this pH gradient, and we could look at various consequences of having more lipid on one side of a membrane compared to the other, and the shape changes and all of the there's some huge changes that result from that. But we got distracted. So we also found that we could load cancer drugs into these liposomes exhibit a pH gradient. And so these are the weak-base drugs that can exist in a protonated form or a net neutral form. Well, the net neutral form can go across the bilayer. If you have a low pH on the inside, it gets protonated and trapped. And so this just shows for doxorubicin, which is a common anti-cancer drug, that you could get so much drug in there that we'd pass the solubility products, and they would get these nanocrystals forming inside this is a 100 nanometer bar here, but inside these vesicles. So we immediately thought, well, wow, this is going to be a good way of delivering cancer drugs more specifically to where you want them. This is an enormous problem, of course. I mean, cancer drugs go everywhere in your body, cause really nasty. Only 0.01% will actually get to where you want it or less. And so if you could deliver these more specifically, it'd be of huge benefit. So we started a company in 1992 called, this is me and 4 postdocs in the lab, called Inex Pharmaceuticals to use these systems to improve the delivery of cancer drugs to tumors. I have to say that this is a great way of keeping a team together. We're still working together one way or another some 40 years later, and we've had some success. I mean, Tom Madden is now CEO of Acuitas, which supplied the lipid nanoparticle for the Pfizer-BioNTech COVID-19 vaccine as an example. But it's been quite a ride over the years. But we started this for cancer drugs, but we soon started working on gene therapies. We hired the CEO, Jim Miller, and one day, about in the middle 90s, he came to me and said, well, I can't raise money for the company putting these old drugs in liposomes. We need to be doing gene therapy. It has much more sex appeal. That's exactly what he said. In other words, sex appeal for investors. And so this really started us off on a whole new path of really, instead of delivering cancer drugs, trying to deliver nucleic acid-based drugs. So let's get back a little bit. So what is gene therapy? What did Jim Miller mean? If you go to Wikipedia, you can find a fairly general definition. There's other definitions. But one version of gene therapy is the therapeutic delivery of nucleic acids into a patient's cells as a drug to treat disease. But delivery is really the operative word. These approaches really require delivery systems. Excuse me, unmodified nucleic acid polymers, they're really degraded very rapidly in the, I got some water here, yeah. They're rapidly degraded in biological fluids. But they don't go to where you want them to go if you inject them. And even if they get there, they can't do anything because they can't get across cell membranes. So, delivery systems are kind of important. Now, what we focused on was delivery of RNA-based molecules to either inhibit gene expression or promote gene expression. So, as you're all aware, DNA goes to mRNA, goes to protein. So the siRNA interferes with the translation by degrading mRNA for a very specific protein according to the sequence in the siRNA. A messenger RNA, of course, if we get that inside a cell, then it'll be taken up into the ribosomes and produce any protein we want. So there's a remarkable statement that comes out of this, and that is that if you can do this, you can potentially treat most diseases, most human diseases, which is really a remarkable statement. But when you think about it, all diseases rely on, they're caused by proteins either being overexpressed or not being available. So both siRNA and mRNA require delivery systems to reach the interior of target cells. So we spent the last 27 years, I guess, developing these lipid nanoparticles to deliver siRNA and mRNA into cells in vivo. So I was going to go through 3 aspects of that, which is the design for encapsulation of these nucleic acid polymers, which is still going on today, and we're still improving these systems. The Patisiran or Onpattro story, which is to treat a particular disorder known as transthyretin-induced amyloidosis, and then I'll briefly mention the vaccine application. So our aim in 95 was to develop a delivery system to try and silence a gene in the liver. We started off with antisense oligonucleotides and then switched to siRNA in the early 2000s. But this is actually one heck of a problem when you think about it, or a challenge, packaging it up, first of all, in a lipid nanoparticle, then it has to circulate around after an IV injection. And say if we're targeting a gene in the liver, then it has to extravasate in the liver. It has to get taken up by hepatocytes, has to then get out of the endosome and into the cytoplasm. And so really building up that capability in one carrier system is not really a straightforward task. And also, of course, you've got the immune system fighting you every inch of the way. That certainly doesn't want foreign genetic material getting into cells in your body. So the first problem that we hit, and this is where the serendipity, some of the serendipity aspects comes in, was that in order to encapsulate negatively charged polymers such as nucleic acid, RNA, DNA, into a lipid nanoparticle, we had to use positively charged lipids. You can see that associate with the negative charge, and we can see the potential then for getting a hydrophobic entity that we could then encapsulate as a lipid nanoparticle. Now, there's a huge problem here, and that is that there's no permanently positively charged cationic lipids in nature. They're really toxic molecules. There's only net neutral lipids or negatively charged lipids. And so we thought, wow, how to get past this one? Because the toxicity, if you're developing a drug, that's always what's going to get you in the end. And so what we did was we tried the lipid that we developed for the lipid asymmetry studies, where we knew that at pH 4, say when we're below the pKa of this tertiary amine, it's going to become protonated, so it's positively charged, whereas at neutral pH, it's not going to be charged, and so therefore, presumably, it's going to be much less toxic. And so we asked the question then, well, can we load these polymers, can we load antisense or sRNA at pH 4, where the DODAP or other ionizable lipids is positively charged, and is it retained when we raise the pH to pH 7.4? And the answer is obviously yes. Otherwise, I wouldn't be giving this talk. So we developed a rapid mixing procedure for formulating these systems. And again, one of the delights of working with lipids, as I mentioned, is they're self-assembling. And so we dissolved the lipids in ethanol, and then through a rapid mixing process, you have to say the sRNA or antisense or whatever else we want to get in there. And one of the first things to fall out of solution is the nucleic acid polymer surrounded by the positively charged lipid. Now, if you do this fast enough, then other things will fall out of solution, such as this PEG lipid here. And you can trap these systems in what we term limit size vesicles, in other words, the smallest size that's compatible with the molecular components. And that when we took the pH back up to pH 7.4, the contents were retained in these systems. And so this really got us past the toxicity issue. And also you can see intuitively how it is that we could get, encapsulation efficiencies that might approach 100%. So so it turns out to be a pretty useful technique. But it's basically dependent on that basic research where we were playing around with the ionizable cationic lipid, and then we had a tool that we could use for making these nanoparticle systems. These are relatively non-toxic. They have a hydrophobic core. This is cryo-TEM picture. Certainly any negatively charged macromolecule, we can encapsulate it, 100% trapping efficiency, scalable. We can change the size, dial the size just by changing the surface lipid to core lipid ratio. There's all kinds of advantages to this process. So we had things that were formulated in lipid nanoparticles. Could they actually do anything? At least we had something that was basically not too toxic, and that was a prime criterion before we even went near animals. So after a conference that I attended in London, this was 2004, I was pursued by, I always term him as a mad Russian because he kind of is, a guy named Viktor Kotelyansky, who at that point was the vice president research and a company in Boston called Alnylam Pharmaceuticals. Now, Alnylam was founded in 2002 to develop a small interfering RNA as a therapeutic. They wanted to silence a particular gene or particular genes in the liver to inhibit production of proteins that were causing problems. And he had the statement was, we have a delivery problem. How do we get our siRNA into the liver, into hepatocytes in vivo? And so this stimulated a 7-year collaboration that went on from 2005 to 2012. We had teams in Vancouver and in Boston, and we synthesized many different lipid nanoparticle formulations. But it resulted in a formulation that we could silence a gene in the liver with a therapeutic index approaching 1,000. In other words, give 1,000 times higher dose than that which we're seeing some biological effect before we saw toxic side effects. So we started off with a question, can these lipid nanoparticle systems silence genes in the liver following an IV injection? So there's really no targeting information on the outside here. I could go into other forms of serendipity. They actually turned out they do target quite well for reasons maybe we can discuss in the question period. So they have a very simple, it's complicated from many drug terms, but we've encapsulated, say, 1,000 of these oligonucleotides in 100 nanometer or 80 nanometer nanoparticle, and we were injecting them IV. How could we, what's going to be the process whereby they actually have some biological effect? Now, lipid nanoparticles get into cells by a process. You're all familiar with endocytosis, the cell-eating process. And so this means that we don't want things to go onto the lysosomes where, say, nucleic acids would be degraded. We have to break out of the endosome at some point so that the oligonucleotide is really delivered into the cytoplasm. And so how can these ionizable lipids destabilize an endosome? What we found was that if we took positively charged lipids and added them to negatively charged lipids, such as those lipids you find in endosomes and other biological membranes, it'll flip the lipid from a bilayer organization. This is the phosphorus NMR signal that I showed at the beginning. It'll flip it straight over into these non-bilayer structures. And we could ascribe this to an electrostatic interaction, changing the shape, as it were, of this molecule and flipping it from a bilayer. But anyway, certainly very membrane destabilizing. And so this took us back to lipid polymorphism. I mean, really basic studies, but it gave us a way forward for designing better and better ionizable cationic lipids. And so we need to design them so that they interact with components of the endosome at some vulnerable part of the endosome maturation process. And so this is our model for saying, okay, what we're trying to do here is to destabilize it by introducing these non-bilayer structures. Now we assess the in vivo potency of these systems using factor 7 mouse model, and this is going to point out some of the amazing advantages of gene therapy approaches. If we can get it to work for factor 7, which is the clotting protein, as you're all aware, then if we can silence that gene, we can silence any other gene in hepatocytes. And so we use this as a model. We had factor 7 the siRNA to silence factor 7, package that in a lipid nanoparticle, introduce that into the intravenously, and then assay for factor 7 in the blood 24 hours later. So we know that factor 7 is made in the hepatocytes, and so if we are inhibiting the production of factor 7, then we're going to see less factor 7 in the blood subsequently. So it's we could do 2 experiments a week. It wasn't high throughput, but we could certainly run through quite a number of formulations. And I should say for nanomedicines in general, screening in vitro doesn't really tell you much. You really have to get to the in vivo circumstance. So we found that the potency of these systems was really sensitive to the species of the cationic lipid, the ionizable cationic lipid that we employed. And so this is just going through a sequence of, we started off with DODAP and KDMA, KC2, MC3, et cetera. They all look pretty similar. I mean, you have linoleic acid chains. We've got a tertiary amine. So why? But there's a huge difference in terms of their potency. And we found that the potency was really very, very sensitive to the pKa, the point at which these ionizable lipids became positively charged. So this is a graph of the potency. Now, potency is 1 over the effective dose, where we could basically knock down factor 7 by 50% as a function of the pKa. This is a log plot here. So you can see if we had a pKa was as little as half a unit away from some optimum, we could decrease the activity by 2 orders of magnitude. So that's really quite a pronounced dependence on the pKa of the ionizable lipids. And so as a result of this, we were able to take effective dose from 10 mg per kg, where we would see some gene silencing, down to 5 micrograms without increasing the toxicity. If anything, we decreased the toxicity. So this is where this therapeutic index of 1,000 came from. Anyway, so at this point, the clinicians, or these, we said, boy, we think this is ready to go into the clinic. And so the clinicians at Alnylam said, okay, what disease should we go after? And the disease that was chosen was a disease that I'd never heard of before, called transthyretin-induced amyloidosis, a hereditary disorder. But as I mentioned at the beginning, by silencing factor 7, we knew we could silence any gene in hepatocytes. So all we had to do was put in a different siRNA that was going to silence our particular target. So this is work that then went on at Alnylam. HATTR is actually a pretty nasty disorder. So the transthyretin, it's a protein that's made primarily in the liver. If there's a mutation, and there are at least a number of mutations, it's a big protein, can cause the formation of fibrils, which presumably deposit everywhere in your body. But they have really nasty effects in nervous tissue and in cardiac tissue. So there's no effective therapy. Usually fatal within 5 years of diagnosis. It's kind of a nasty disease, as is indicated here. It's a wasting disorder that goes through the stages, as indicated here. Kind of particularly nasty because it's hereditary, and so if you find you're stumbling in your 30s, you sort of know what's in store for you because you're likely to have seen a relative go through a similar process. So this is where the simplicity of gene therapy approaches comes in. Say, okay, well, this is caused by the protein transthyretin. Well, this is stop it being produced, and so this is taking a lipid nanoparticle now containing siRNA to silence transthyretin, and the idea is if we shut that down, then we're going to reduce the circulating tetramers, the deposition of the fibrils, and perhaps we might even get some clearance if there's very little in the blood. So potentially a simple solution to what is really a devastating disease. This is just the results of the phase 3 study, which came out in 2017. So there was 148 patients who got the drug at a dose of 0.3 mg per kg IV every 3 weeks, or they got sterile saline as a placebo. And the neural impairment score was a primary endpoint. Quality of life, weakness, et cetera, were secondary endpoints, ability to walk, and so on. Now, so these were some of the top-line results for the neural impairment score. So over the course of the 18 months, the patients that were on the sterile saline, or the control, were getting steadily worse. Their neural impairment score was getting higher. Whereas those patients that were being treated with the drug to silence transthyretin in the liver, if anything, were getting better. So really improving the status of these people suffering from a hereditary disease, and quite a remarkable thing. So the phase 3 trial results were announced in 2017, as I said. I also refer to the p-value for the neural impairment score. This is 10 to the minus 24 1 over Avogadro's number. They're absolutely sure that this drug works, but it's equally convincing for self-reported quality of life, muscle strength, ability to walk, nutritional status, et cetera, et cetera. So it's certainly one of the most, that's the most impressive clinical trial results I've ever seen. It really points out how these genetic drugs, they really work, because you're going right after the root cause of things. So this was approved by the FDA, the trade name is Onpattro, by the FDA in 2018 for treatments, the first FDA approval of a siRNA-based drug, and a clinical validation, obviously, for the lipid nanoparticle delivery approach really dramatically demonstrates the power of the gene therapy approaches. Okay, I'm just going to go on now to the vaccine story, which really took place between 2012 and 2020 for the COVID-19 vaccine. Now, this is when Onpattro went into the clinic in 2012. Of course, there was no point. There's nothing you can do as a scientist that has that point. I mean, it's all the clinical trial. You can't change things in the middle of a clinical trial. And so we said, well, okay, if we can deliver siRNA and get that to silence a gene in the liver, it's a pretty big molecule. It's not as big as an mRNA, but maybe if we package mRNA in the same way, we'll get some gene expression. And so that's what we did. And what we found was that we could use the lipid nanoparticle approach to use the liver as a bioreactor to produce any protein we want. So think about that. If you want to win the Tour de France, this is the injecting mRNA coding for erythropoietin, and you get, this is a pig model, you get super physiological levels of EPO and the corresponding increase in hematocrit, et cetera, in these animals. So this points out this was our whole focus was why there's just an enormous number of diseases, rare diseases, et cetera, that one can go after using this approach. But we again had another serendipitous event because we were approached in 2014 by Drew Weissman of the University of Pennsylvania. He'd been working with Katie Carrico, and Drew Weissman and Katie Carrico are co-winners with me of the Gairdner Award. So they'd been working for many years to try and reduce the toxicity of messenger RNA, the immunogenicity. And so they come up with, they'd shown by modifying mRNA they could beat that problem, but still they had a delivery problem. How do we get mRNA coding for viral proteins into muscle and immune cells in vivo? And so they needed a lipid nanoparticle, obviously enough, to get the mRNA coding for vaccine into the cytoplasm of both, say, muscle cells and antigen-presenting cells to get a vigorous MHC1 or MHC2 class immune response. So the first thing that we looked at or that Drew looked at, was Zika virus. So the lipid nanoparticle in this case contained messenger RNA coding for a surface protein on the premembrane and envelope glycoprotein on Zika virus. And so this was in a mouse model and just showing that this is intradermally injected lipid nanoparticles containing mRNA coding for the Zika virus, the premembrane and envelope glycoprotein, a bit of a mouthful, and then challenging it 2 weeks or 20 weeks with the Zika virus itself. And the quite remarkable results, in as much as was published in Nature in 2017, complete protection both at 2 weeks and 20 weeks against a Zika virus challenge. So this is looking at the viral copy numbers per milliliter And so these things turned out to be really good vaccines. I mean, this is not a direction we were going in at all. It was only, we were trying, as I say, express proteins in the liver. You get a phone call, and then, things change. Now, this caused Acuitas, it caused us to start working with the BioNTech to develop an influenza vaccine. Obviously, that's from a commercial point of view of huge interest. Now, it turned out that BioNTech had also been working with Pfizer on the flu vaccine. And so in January, February of 2020, all those efforts were switched using the lipid nanoparticle that we'd been supplying to developing a COVID-19 vaccine. And you all know the results of this, that in November of 2020, this vaccine was demonstrated to be 95% effective against COVID-19, independent of age or gender, race, etc., and was reasonably well tolerated across all populations. This is 43,000 participants, so this is certainly one of the larger clinical trials you're ever going to see for a vaccine, remarkably convincing results. They expected to produce 1.3 billion doses by the end of 2021. They were much closer to 3 billion, I think, and obviously it's been approved in many jurisdictions. And so that's really is to say, we spent 27 years developing these systems. It's been a pretty amazing journey. I hope I pointed out how that journey has really depended on some very basic research and also some good luck, the serendipity. Really quite remarkable. But the point is, it's just beginning. I often refer to mRNA systems as really being, the third generation of pharmaceuticals, where the first generation small molecule drugs, second generation biologics, monoclonal antibodies, etc. And then third generation, these gene therapies for protein replacement. You don't have a protein being made. Okay, well, let's make it. As vaccines, as is obvious, gene editing is another approach that we can use to alter gene expression. So quite remarkable how the broad applicability. And the other thing that's kind of mind-boggling, as we're used to, developing a drug that might take 15 years and $1 billion. Once we've identified the protein that we want to express or silence or edit or whatever, we can produce the siRNA or the mRNA in a time on the order of, say, a month or so, and we can package it in a day or so. And so you have this really highly targeted, personalized medicine available in a time on the order of weeks. So this changes the paradigm of medicine as far as I'm concerned. It just makes a huge leap. So we're going to see a multitude of these approaches coming in over the course of the next few years. Obviously, other vaccines are certainly in the cards. Universal flu vaccine, for example, HIV is looking promising. The chronic diseases, for example, heart disease. There's just a paper published basically for heart failure, showing that you can eliminate fibrotic lesions in the liver and the heart gets better using a CAR T-cell approach, actually. Inherited diseases, obviously replacing a protein that's not being made. Anyway, the list goes on. And so it's a very exciting time to be in medicine when we're suddenly enabling all of our understanding of molecular biology in a very direct therapeutic manner. So it's changing the game in a fundamental way. Now, I can't stop without acknowledging all of the people I've worked with for literally 40 years, as I pointed out, as well as many, as Mick Hope, Tom Madden, as many such as Steve Ansel, Ying Tam, Barb Ngui, Paul Lin, 20 years or more. The people at Arbutus, Ian McLaughlin, James Hayes, Al Nilam. This is a hugely successful collaboration. Mark Tracy, Kenneth Kink, Martin Mayer, Manna Manoharan, Marco Cifaloni in the chemistry department at UBC, who really played a big role in breaking the whole problem of the ionizable lipids. These things work in the brain. I haven't discussed that, but in my own group, and of course Drew Weissman at the University of Pennsylvania, So with that, I'll close and take any questions that people may have.
Thanks, Peter. That was a fantastic talk. And if there's anybody who has any questions, please raise your hand. But maybe I'll start with, silencing a protein is one thing. Putting one in that's going to be upregulated, how do you control that regulation? How do you know you don't overexpressing a protein? And have you even thoughts to that?
Yeah, I mean, one of the things about this approach, of course, is titratable, so you can start low and work it up. The other is that it's not like you're inducing production of a protein forever. You're going to get it going for maybe a week or two, but that's about it. And so it's self-limiting in that extent too. So if you're causing nasty side effects, then okay, stop giving it, and then other things will.
So that would be
normal.
My next question. How often do you think you'd have to give something if you were dosing?
Depends on the length of time that you want, and also the stability of the protein that you're producing. Like antibodies, we can make those in the liver, say if you put the heavy light chain mRNA in, and they'll circulate around for, say, a week or so, because they're fairly stable. Other molecules, might be, a matter of an hour or so, and like there. So it's going to. These are areas where you're going to see improvements in the technology itself. More of a depot effect to have things play out over a longer time. There's a long way to go in the ways in which we can manipulate things.
And in your delivery system, do you think there's going to be a way to actually target it to different tissues? So right now you've been talking about like getting it into the liver, but can we actually restrict which tissues?
It's going to be tricky. That's going to be tricky. People have been trying to target these lipid or any nanomedicine actually more specifically to where you want it, and then cancer cells is an obvious choice without any success over 40 years of trying, right? So, and that points out how difficult it is. You tend to raise an immune response or anything you put on the outside, and you get aggregation, they're difficult to manage, but this goes on and on. I've worn out 5 graduate students on that project. The last one refused to go on unless I changed your projects. Okay, I did that. But it points. But there are other ways to go. For molecular biology, you can start to say, okay, it won't be expressed in a particular cell. That requires pretty sophisticated molecular biology, but I think is doable. Other approaches are let's get things that are activated by external stimulation. And one of the nice things you can do with lipid nanoparticles, we can incorporate things like iron oxide nanoparticles or gold nanoparticles, and then laser or RF or whatever other stimulation. You can see ways of saying, okay, turn something on here, but not here. So there's going to be approaches that will achieve that.
Okay. Any questions from the audience? A question here, and then a question way at the back and up there. So we'll start here and go up there. So please, loudly.
All right. Very nice talk, Peter. You sort of alluded to that a little bit, but with mRNA delivery, it's obviously a time-limited expression, so that works well for an acute disease or a vaccine. But have you worked on DNA delivery systems, say, compacted with protein for specific integration for, say, an inherited chronic disease where you don't have that delivery?
Yeah, we worked on it without any success.
It's commonly used in cell culture for transfection.
Absolutely. In cell culture, you've got rapidly dividing cells, and so that but the problem is that for cells that are not rapidly dividing, and so therefore the nucleus isn't, you can't get things into the nucleus as readily, then these systems don't work. And so that's which is, we certainly haven't beaten that one yet. We're feeling pretty good about having got as far as we've gotten, but yes, there's other barriers to get through for sure.
Yeah, I guess that DNA moving to the nucleus is a big problem because I think it gets degraded.
Yeah, and there's no doubt we can get DNA into the cytoplasm in the same way that we can messenger RNA, but to get it into the nucleus, we haven't achieved, at least for non-dividing cells, we haven't achieved that. Oh yeah, no, we're fiddling around with every possible, we really haven't figured out. It's coming to light now a little bit, but are the structure-activity relationships between the structure of the lipid nanoparticle and the actual activity in vitro and in vivo. So those things are just starting to become elucidated. It's quite remarkable. You have a very successful medicine, and we still don't understand some of the basics of how it actually works. But those things are becoming clearer. So. And a lot of it has to do with manipulating the various components that we have in there.
Yeah, thank you for the talk. So my question is, in case somebody has to take, for instance, somebody has to take COVID-19 vaccine, universal influenza vaccine, malaria vaccine, Zika virus vaccine, and possibly other kind of vaccine that are made with the lipids and particles. So are there limits of the amount of lipid and particles somebody can take in at a time? Or can all this vaccine put in one particle? Or, so
I mean, are you asking whether you can use the same lipid nanoparticle for all those different vaccines?
So if it's used for different vaccine, and somebody has to take these vaccines at the same time, so are there limit of the amount of this this lipid particles that should be in the biological system at the time?
Oh, I mean, it looks like you can use the same lipid nanoparticle to deliver whatever mRNA for whatever vaccine, and it works just fine. So and this is where the lipid nanoparticle that's been used for, say, the Pfizer-BioNTech vaccine is going to find the exact same lipid nanoparticle is going to find a lot of application for other vaccines, particularly now it's been tested in 3 billion people. So it's but yeah, you can use the same delivery system for quite a variety of mRNAs.
Some question down here.
Thank you for the nice talk. So I have a question. So you said the mRNA vaccines, and the liver as a bioreactor. So liver is the bioreactor where there is you are targeting mainly the liver, and it's composed of several cytochrome P450. So in a patient, like COVID-19 patient who has compromised cytochrome P450 enzymes, and utilizing liver as a bioreactor, how would you
Well, yeah, well, I mean, obviously for the vaccine, we're not using the liver as a bioreactor. We're injecting that intramuscularly, and it's antigen-presenting cells and so on that are producing the vaccine response. Your question as to how, say, if your liver is compromised in any way, whether you can use that as a bioreactor more effectively, we don't know the answer to that yet. I mean, it hasn't really been tried.
So compared to liver, the lungs in the cytochrome P450 enzymes are fewer compared to liver. So why not to target lungs where there is the virus load is more compared to liver?
Yeah, this has to do with, as I think I alluded to at some point in the presentation, we get a natural targeting to the liver. I don't know if I mentioned it here in the previous lecture, but when you eat a fatty meal, it gets packaged. The fats get packaged into chylomicrons, and they go around your body. And anyway, they come back to the liver as chylomicron remnants, and they adsorb a lipoprotein called ApoE, and that triggers uptake into hepatocytes by the LDL receptor. Well, it turns out that our systems hijack that process. They adsorb ApoE, and it gets taken up into the liver that way. So that's we're taking advantage of a natural phenomenon.
Jude?
Yes, I guess the question over there was asking about toxicity, whether there is kind of toxic impact of having multiple of this. And I guess as an immunologist, then maybe I can ask, is there a possibility that you can use, you can have multiple antigens included in the same nanoparticle?
Yeah. Oh, yeah. I mean, with regard to toxicity, I mean, if there are some people say have a response or a reaction to the PEG lipid or the polyethylene glycol that is in these. There's some evidence that some small percentage might. I should say the overall reaction to these systems is not really any, in terms of adverse events, is not really that much different than any other commonly accepted vaccine. So it's, there are, you'd expect, even if it was a different vaccine for a different purpose, that they would see the same reaction if it's to one of those components. With regard to the multivalent, or they're putting in different, this is, there's now experiments being done on this would be a flu vaccine that is basically going to be effective against all strains of flu. And that's just accomplished by putting in every protein you can think of in the flu vaccine, into one nanoparticle, and then getting an immune response that really is very reactive to all sorts of components of the virus. So it's still early days to see how well these things are going to work generally, but they're highly flexible in terms of you can package, there's really no limit to the amount of mRNA you can package into these systems.
I think non-liver targeting is an active area of research now. So for some companies like Precision Nanosystem Innovation, they are making new lipids or changing the composition of the lipid to achieve non-liver targeting. Where there is academic papers that are integrating the nanoparticles with protein, antibodies, peptides to achieve non-liver targeting. So what is a better way to go, manipulating the lipid composition or decorating with proteins?
Yeah, some of those studies which are getting to different tissues have really ended up using lipids that are a bit toxic. So I'm kind of wondering how well they're going. Anyway, the jury's still out on how it is you might get things to one tissue versus another. I mean, the approach we're taking is to say, okay, your first thing, if you want to transfect, say, bone marrow or whatever, lung, is to actually get transfection there. So with the current systems that are used, say, for liver delivery, it's all over in 30 minutes, and 90% is in the liver. So it doesn't get anywhere else. There's no chance of transfecting other tissues. So then the trick is to say, okay, well, let's get long circulating systems that do get to these other tissues. They might still transfect the liver, but maybe we can get a more general gene expression throughout a number of tissues. And that's the direction we're going in now. And then the trick is, okay, let's turn them on and off in particular locations, which is kind of a question that you were asking. But the very first thing is to get the material there and get something happening. Then you can worry about the fine details of getting things expressed in one tissue versus another. It's going to be very difficult to target two particular cells. That, as I said before, is a tricky thing. But if you can actually does it really matter if you express the CFTR protein in other tissues? We don't know the answer to that yet. If you're going after cystic fibrosis. So these are questions that just remain to be answered.
Well, with that, I'm going to bring our session to a close and really thank Dr. Cullis for a fantastic talk. I think you really gave us the history of going from being a physicist to in a department of biochemistry, and really the bridge was the technology, and then how you've just gone with the science and let that direct your direction over the years, and how what we think we are doing, at one point we don't see its utility, but then it comes into its own in a number of years. I think it was a great story and a lot of lessons to be learned in the talk. So thank you very much. Fantastic time.
Thank you.